There's what's called hydrogen bonds, although they're not really bonds. They're more just like magnet interactions of wanting to stick to each other. So that means water ends up being this sticky, self-sticky kind of thing, and it can also stick to other stuff. So it can stick to itself, which is called cohesion, and it can stick to other stuff, which is called adhesion. And those two things make a tremendous difference in how water behaves. So, water is really weird. It does really weird stuff. Like, almost every other fluid, when you freeze it, it actually gets compacted. Water, when you freeze it, it expands, which is just weird. Yeah, totally weird. Because of the sticky nature of water, because of the way that each of those positive and negative ends interact with each other, water is able to hold a lot of heat. So some nonpolar fluids, you heat them up, they heat very quickly, volatilize very quickly. Water volatilizes very slowly. I mean, if you think about it, it's like the water molecules are moving around inside them, and every now and then, one of them is moving so fast that when it hits the surface, it pings off and goes off as a gas. That's not that common, and when it goes off as a gas, it actually pulls the energy that's in that moving molecule with it. So it takes a lot of energy to get the molecules of water moving enough to be able to start kicking them out. So there's actually quite a bit of energy in the motion of water that can be utilized to move things along with the cohesion and adhesion. So, all right, water's weird. We have to also remember that there's a specific kind of osmosis that deals with water, a specific kind of diffusion that deals with water. And that is that because of the sticky nature of water, when water is complexed with some other compound, usually ions, it's essentially out of the picture of the molecules of water moving around. So think about it, right? You've got a whole set of water, and there's molecules moving around, and they come in contact with a charged particle, and they'll form a sphere of water, of each individual molecule of water stuck around that ion. Those water molecules that are stuck around the ion are not counted in the molecules that are moving around. So when you add stuff in when you add ions into water, and you have an equivalent number of molecules in a separate container, and you don't add something in the one that you've added the ions in, there are functionally fewer molecules of water moving around. Between the two of those things, that sets up the equivalent of a diffusion gradient. There are more water molecules in the ones that don't have the ions in it moving around. Same number of water molecules, but some of them are complex with the ions and aren't moving around. So what would happen if those things had a membrane between them? You'd have lots of water moving around in one and less water moving around in the other. What would the diffusion gradient for water make happen? What's that? Well, which way would it go? Would it go to the way that you have nothing added in, or would it go to the way that you had ions added in? If water's going to move back and forth, it would go to where the ions are, because there are fewer molecules of water moving around. We call it free water. So free water is water that's not in hydration spheres or not complex with other ions. So if you were to have this semi-permeable membrane between places where there's lots of ions and very few ions, water would diffuse, or because it's water, we call it osmosis, across the membrane from the locations where there are very few dissolved things to the locations where there are lots of dissolved things. And it would keep on moving until you ended up with the same amount of free water on either side, the same number of molecules on either side. So, also, that's actually an energy gradient. If you can put ions into water and create water movement from one way to another, you've actually set up an energy gradient that moves those water molecules from one place to another. The easiest way to remember this is just that water follows the solute. Right? The place that has more stuff dissolved is where water is going to move to. So we call this water potential, and it's a measure of free energy in each of those each of those types of fluid. So what plants are able to do is utilize that free energy difference. So the process of moving water through the plant body is called transpiration. And what does it is the fact that the shapes of the cells in xylem are like little straws. Has anybody ever seen like capillary action where you stick a little straw into water and it's like the smaller the straw is, the farther up the water will go? That's because water is adhering to the sides of the straw, and cohesion to itself pulls more water up. So you actually get this little lift of water coming up. There's also something too about pressure on the side, pushing you up, but whatever. So with evaporation taking place at the tips of the leaves, at the stomata of the leaves, think about this, right? There's water moving away. There's water evaporating away. So it's actually creating a decrease in free water at that location because of the evaporation away. And that evaporation away with this loss of water at the leaf surface sets up an entire chain reaction running through free water potentials through the rest of the plant body that actually moves water through it passively. So there's a bit of capillary action pulling water up on things, but at the same time there's free water differences between the whole column of water. And that makes it so that water passively will move from outside of the roots, into the vascular system, up the xylem, out to the leaves, and evaporate away. It's not pumping water through. There's no physical action of the plant making this happen. It happens passively, simply because of the facts of physics. It almost like, right, how could water flow uphill? It flows uphill because of these free water potential differences throughout the entire throughout the entire plant. So we've got the point where all of this is taking place is the stomata. In order for transpiration to happen, you have to have water evaporating away at the leaf surface, which means you have to have your stomata open. So when we had talked about stomata previously, we were talking about it mostly in terms of gas exchange, right? When it's open, you're able to get carbon dioxide in and oxygen out, and you can, as a plant, and you can photosynthesize continuously without having photorespiration, mess things up because of the competition of CO2 and oxygen for the binding site in rubisco. I know that was a mouthful, wasn't it? So, with the setup of leaves, you've got a loss of water here, which gives us a deficit of water molecules in this space. So to make up those deficits in the water molecules, water moves out of the vascular bundle and into the space where it's been evaporated. But because of the deficit in the vascular bundles in the leaves, water is moving to take its place from the xylem. And because of the deficit in the xylem, water is moving to take its place from in the root. And because of the deficit in the root, water is taking its place moving in from the spaces outside of the root. It's essentially just the math of free water potential at each of those locations. And in the locations where there is higher amounts of free water, it will move from those locations to the places where there's lower amounts of free water. So think about it, right? Water is evaporating away, which means very low free water. So places in the root where there's lots of water, or places in the soil where there's lots of water, few dissolved ions comparatively. So really what makes this run is simply the difference in free water potential at each part of this process. at each place in the organ. Yeah.
So when you're when you were talking about how like water potential difference in capillarity and then evaporation, is the capillarity and evaporation helping this, or are they
Yes.
parts of this?
They're parts of it and helping it, right? So the way that water will stick to the sides of a tube and sort of work its way up, that's a capillary action. That actually makes this process happen a little bit easier because water is already adhering and pulling to the sides of the capillary. Think about how tiny each one of those little xylems is, right? Little tiny xylems. That makes it so that with free water potential low at the high end of the tube, capillary action gives it a little pull on the side, but with free water missing up here, it essentially flows to that location, right? The same way in osmosis, it flows from locations of low dissolved stuff to high dissolved stuff. That's a measure of free water potential, what's dissolved in there. So those things can come from differences of what's dissolved, that's solute pressure, differences in pressure potential, that's like what's pushing on the water from air pressures basically sitting on it, and the differences of water potential differences of what's dissolved or what's free water. And all you got to do is recognize that from soil to root to stem to leaf to air is this progressive loss of free water potential, progressive loss of whatever component of it you're measuring. It doesn't matter if it's necessarily from dissolved stuff or if it's from pressure differences. What ends up happening is that there's a net flow of water from the soil through the plant body and out the leaves. It's a remarkable process, and it's totally passive. That's the really cool part, is that the physics makes it run, not muscles pumping a heart moving the thing through. It's not pumped, it just happens, provided there's a sufficient amount of evaporation taking place at the leaf, provided that there's this continuous decrease in water potential as you move your way down, as you move your way up. And that's it. It is really amazing that this has evolved. So when we talk about vascular tissue and how important vascular tissue is, this is what vascular tissue evolved with. Right? Vascular tissue is the ability to utilize this physical process to move fluids throughout the plant body. Yeah.
How did that? I'm just like trying to picture. How did the vascular cylinders? How did they evolve from like they didn't have it at all, and then all of a sudden, like the water potential shaped the plant, or
you can see it in things like ferns. They don't have a vascular cylinder within their roots and stuff. They've got a different structural makeup of the way that those vascular tissues are. I mean, pretty much if you can think about any, in an evolutionary sense, any organism that can move further away from water, get their leaves better exposed to sunlight to photosynthesize better, has an advantage. So I can imagine it happening incrementally, where if you can just get it further up in your plant body, you're better off than something else that can't, right? So the evolution of this is really just cells where the cell walls were modified to be thicker and then remove the cellular material inside, and you've now got a straw that can help with capillary action, bring it up. So remember, this is also, we also talked about the evolution of leaves, stomata, a bunch of other things too. They're all interacting with each other for these systems to become more and more complex. Those early stages like Psilobium, right? Psilobium doesn't have leaves, but it's got stomata. It's got little holes. So this process actually evolved before the development of leaves, the ability to pull fluids up through evaporation at the surfaces of your stems, right? Leaves were a later development. But now that you have leaves, you have more surface area for evaporation to take place, so that means the bulk flow can happen a lot faster. That means your body, the plant body, can get bigger. So, make sense?
Yeah.
Right? The thing about evolution is that that any minor improvement over not having it is selected for.
So
Even though we look at this integrated, intricate system, any part of it that evolved that gives an advantage would be advantageous to not having it, and it would be sticking around. This actually links up with something else that we're going to talk about, because vascular systems don't just move stuff up, right? This is what drives things up. Vascular systems can also move things down. We'll talk about that in a moment. So what you're thinking about here is just bulk movement of water, just differences in water potential from exterior to the plant, into the root, into the stem, into the leaves, and out the rest of the way. It's simple physics. But part of this process is bringing things into the plant that it needs. So we've talked about this being simple physics of moving fluids through. But in reality, there is a single spot that is the location where different kinds of minerals get selected to be brought in or not. So it's not just bulk flow, whatever happens to come through that fluid is what the plant needs. Every water molecule that comes into a plant body has to go through at least one cell. An endodermal cell, in the endodermis. I mentioned this last time. But conceivably, a water molecule could move in from the soil, this is simply because of differences in partial pressure, into the spaces between the root cells, but it has to go through one of the endodermal cells, because there's this connection between the individual endodermal cells called the Casparian strip. The Casparian strip here makes it so that it cannot circumvent going through a cell at the endodermis. Every molecule of water has to be brought into the endodermal cell. And that means that proteins on the surface of those cells can help regulate which kinds of molecules or which kinds of elements come in, what dissolved materials can come in. So what actually makes it into the plant body, what actually gets brought to each of the cells, mineral-wise, happens at the Casparian strip. It almost like filters everything that's around in the water outside and selects the things that are going to be beneficial to the plant or that have cell transportation systems that brings that in. And then after it moves into that cell, that water molecule could conceivably go back to the spaces between the cells in the vascular cylinder and work its way up through the same passive processes and differences in water potential all the way up the stem, all the way out into the leaves, working its way through the spaces between the cells and out the stomata, never actually going through another cell in the entire time through, except for the Casparian strip, limiting that water molecule from moving around the endodermal cells. It's got to go through the endodermis.
So does the Casparian strip, is it the filter, or does it just...
The Casparian strip is connections between the cells of the endodermis. So the strip itself is not the filter. The cells are the filter. The cells are the things that actually determine what moves through.
So it makes sure
it's got to go into the cell and then into the vascular cylinder. It's a block from making it so that things can't go around the cell. It's got to go through the cell. But no other point in the entire process of plants does anything actually have to go through the cell. It can go all the way up through, or at least water molecules goes all the way up through the rest of the plant body, simply from the processes, the passive processes of differences in free water potential.
And the Casparian strip is only in the endodermis.
Casparian strip is only at the endodermis. So imagine, think about how important this thing is for the plant, right? This one little circular set of cells in the middle of the root is really what determines what actually gets into the plant from the root system. So all the minerals that a plant might need, all of the things that like dissolved ions, all that stuff that the plant needs, goes through a cell at the endodermis before moving into the rest of the plant. Pretty cool, huh? So, how does the plant regulate this? This is also done passively. This is not something that happens because the plant has a nerve system that can recognize what's going on and have a response to it. What happens is that it's simply a matter of how much water is actually making it to the cells around the stomata. If the plant is fully hydrated, that means that there is water moving into the cells, and there's you ever seen like when you take a stalk of celery that's all floppy and starting to get wilted, stick it in a glass of water, it gets stiff again. So the way that plants are set up, water will flow into individual cells, and the pressure on the inside of the cell pushes against the cell wall, making the cell be stiff. If it's got too little bit of water, the stiffness of the cell sort of diminishes. So what happens with stomata is that there are these cells that are alongside of it called guard cells, and you can think about a guard cell like like the balloons that are made by clowns and stuff, like the balloon animals and things. You know how like you fold the balloon animal back on itself and you get this little. If you have the balloon animal, you make like a little twist and you get this thing that looks like this, right? If you allow those to be filled with air, you'll actually see it twisted and it sort of opens like this. And if you let the air out, the pressure that's being pushed onto it allows this to come in and leak and go up against itself. And it's actually the exactly analogous process. When the thing is filled with water, when there's plenty of water pressure, the sides of the guard cells pull apart, opening the stomata. So the plant has to be fully hydrated to have the stomata open. If the water is low, so we're talking about it in terms of water potential here, the things fold back in and close. So you can also realize that this happens with the plant being hydrated or not, but it can also happen with the plant regulating the ions in those guard cells. So what would you expect to see happen if ions were moved into the guard cells? Where would water go? It would flow in and open the things up, right? And if ions were pulled out of those cells, what would happen? It would close back up. So the plant can actually recognize what the conditions that it's in and give a physiological response to either open or close the stomata. When it's fully hydrated, it's no big deal. All the water's flowing in, things open. If it's low in hydration, the thing will close passively on its own. But the plant can also alter that by adding or removing ions into those guard cells. So the ions that it uses is potassium, one of those important limiting nutrients for plants, right? They've got to have enough potassium to be able to do this. And it's simply a matter of turgidity. Simply a matter of how much pressure is pushing against the cell walls. High pressure, the guard cells bow out. Low pressure, they fold in and block it. So when conditions are unfavorable for transpiration, they'll close and when they're favorable, they'll open. So when we were talking about CAM plants, right, that are or C4 plants that are circumventing the problem of having your stomata closed, if there's too much evaporation going on at the leaf surface and not enough movement of water up to the leaf surface, there's a deficit of water, and so the stomata close. If you've got closed stomata, it allows the chance for photorespiration, and that's a problem. So it's evolved this other way of getting carbon dioxide when the stomata are closed. So it can still photosynthesize even when the stomata are closed. So you can see just how important this thing is, right? This is incredibly important for the functioning of the plant's system. So that's dealing with how do you get stuff from the roots up to the rest of the plant. But remember, the plant is making its sugars at the surfaces where photosynthesis is taking place. For most plants, that's leaf surfaces, although many do it in stems. How do you make your roots grow if your only place where you're making sugars are at your leaf surfaces? And you need to get that food to the cells in the root. Well, you have another set of cells that are specialized for doing that. So xylem are the cells that will get water moving from the roots up to the leaves. Phloem is the cells that get those fluids that are heavy in sugar at the cell surface or at the leaf surface down to the rest of the plant. So the cell types that do movement in xylem are called vessels and tracheids. The cells that are doing it in phloem are called sieve tube members, and they have a companion cell. So the reason why they're called sieve tube members is you'll see between each individual cell are these little sieve plates that look like this. This is actually a scanning electron micrograph. And it's a cell wall that's perforated. And the cell wall of one sieve tube member sits right up next to the cell wall of the next sieve tube member. In xylem, the cell wall of one vessel and the cell wall of the neighboring vessel dissolve away. So it's just the exterior cell wall. It's forming what's essentially a single tube. In the sieve tube members, you have these little plates at each location. And they do weird things like the flow, this is all blocked up dissolved matter, and at a certain point, sometimes sieve tubes get blocked, and then a new one grows around it. And so, Dr. A can tell you all about it. It's a really cool process. So, all right. In order to get sugars from the places where they're made to the places where they're needed, they don't just flow passively. Well, they do. The flow is passive. We'll talk about that in a second. But the action of getting the sugar into the phloem doesn't just happen on its own. There are active transport systems that actively move the sugars into the phloem cell, into the sieve tube members. And that's why they've got these little neighboring cells that are called companion cells. This is the shot of a companion cell running down next to the sieve tube member. Because the companion cell is loading and unloading that sugar in. It's moving it into the phloem or moving it out, depending on what's needed at the different places. So this movement of sugars is called translocation. So this still happens passively. The flow happens passively. The movement of sugars into and out of the sieve tube members and companion cells happens actively. So this sets up a weird paradox. How can a plant be moving fluids against gravity passively while at the simultaneous time move fluids in the opposite direction passively? That's a weird paradox. They're actually coupled together. They're coupled together in this process. because the movement of one influences the movement of the other. And this is the model that we think does it. It's called Munch's pressure flow hypothesis. And there's two things going on here. So the way that these two processes are coupled is actually pretty cool. So we've already talked about what happens with xylem, right? Pressure flow differences or free water potential differences across the whole plant that is moving water in one direction. That sets up the chance for it to be coupled to the other process. So the number one thing you got to remember about Munch's pressure flow hypothesis is that transpiration has to be going on for translocation to take place. You have to be able to be moving water through the xylem for you to be able to move the sugar-loaded fluids through the phloem in the opposite direction. Okay, so how does that work? Well, we can think of it this way. We're going to go with xylem has got water flowing on its own just because of what's going on with transpiration. But at the root location, there is a need for the sugars. So the plant is utilizing those sugars to grow and provide cells. That, every time one of those sugars gets used by a mitochondria to form ATP, that sugar is now gone in the mix of what's dissolved in that area. And that creates a deficit of sugar at the cells that are actually using it for growth. At the places where the sugars are being made, photosynthesis is taking place, they're making sugars. Every single time a new sugar is made, you now have a higher concentration of sugar. Right? So you end up with a concentration gradient running from the source, the cell that's actually making the sugar, has it in a high concentration, gets loaded into a companion cell, and the difference between the high concentration in the phloem next to a companion cell near a source and the difference in the concentration of sugars at the companion cell next to the sink makes for a gradient of differences in the concentration of phloem. So what would you expect to have happen with actual sugar molecules across this gradient of concentration gradient of sugar? From simple diffusion, right? Just setup of diffusion, there would be a bulk movement of sugar from the locations of high concentration near the leaves to the locations of low concentration near the cells that are actually utilizing it. Okay? So, when somebody tried to calculate how long that would take for an individual sugar molecule to move from the source to the sink, it was on the order of several years. Right? Diffusion, even with this big difference, is not fast enough to supply those cells that are growing with enough sugar to actually keep them growing. Simple diffusion, while setting up a bulk movement of ions from source to sink, is not enough on its own to get all of the cells that need it supplied. But if we add in the fact that xylem and phloem are right next to each other, you guys know this because you have seen it every single thing you look at. Vascular cylinder, what do you have in the dicot? You've got a little star of xylem and phloem sitting right next to it, right? Vascular bundle of monocots. You've got those xylems and phloem right next to it. Vascular bundle of dicots. You've got the bundle, you've got the section of xylem and procandium and phloem right next to it, and fibroblasts, right? The dicot root. Even out at the tippy tip of the littlest leaf that has a little vein in there, there's xylem and phloem right next to each other. That is not a mistake. That is a structural, fundamental setup that allows this to happen. Because if you start thinking about, we're going to throw in osmosis here, right? We're going to start thinking about if those two things are next to each other, there should be movement of water from xylem to phloem or phloem to xylem, depending upon what the dissolved stuff in each of those are. So think about it, right? We've got companion cells actively loading sugar molecules into phloem. But the phloem is right next to xylem. And xylem has water that's moving up from the roots that doesn't have much stuff dissolved in it. What would happen because of the difference in the free water potential between phloem and xylem? Water would move from xylem into the phloem. Okay, so now you've got an increase in pressure because water is moving into the phloem. That's actually like Turgor pressures pushing against the sides of the cells. Okay, now at the bottom or at the source or at the sink, you have locations where the companion cell is actively pulling those dissolved materials out of the cell. And in fact, the dissolved sugars have to get down to a level that's below the dissolved minerals from the root that have been picked up from the root. And once that happens, there's now actually a higher concentration of dissolved materials in the xylem. What would you expect water to do from the phloem cells that have had their sugars removed and the neighboring xylem cells that have a bunch of dissolved ions from? Water would move the other direction, right? So water is moving into the companion cells near the source and out of the companion cells and phloem cells near the sink. You have set up a diffusion gradient, but you've also set up a pressure difference. You've also set up high pressure where water is moving into those phloem cells, and low pressure where water is moving out of those companion cells. And what happens with water between high and low pressure, or anything between high and low pressure? It flows from high pressure to low pressure. There's actually a flow of movement from the locations near the source to the locations near the sink through the phloem because of this packing and unpacking of sugars, and because of the diffusion, because of the diffusion of high sugars to low sugars, and because of the osmosis of water moving out of the xylem at the source or at the sink and into the xylem at the source, or into the phloem at the source. So, why would this shut down if the xylem wasn't moving? Why would this system stop working if xylem wasn't still actively moving fluid? What's that?
Would there be too much sugar in the phloem?
Well, what happens if osmosis is allowed to go between two things, right? It would get to where you end up with even water potential in both sides, right? So if this isn't moving, and if this isn't moving, you would end up having a point where these guys all became equal. If it's not continuously flowing in the xylem, the chance to even out between the xylem phloem source and sink happens, and flow stops. But if you have continuous flow in the xylem, then the chance because what you'd have would be ions would start to osmose this way, would start to diffuse this way, ions would start to diffuse this way, and you'd end up with the whole entire thing equal in its dissolved stuff. But with xylem continuing to flow, you end up having the chance for water to move into and out of it. So you get dissolved stuff evening out. You get the pressures between the two xylem and phloem evening out, and there'd be no bulk flow of water from one to the other. There'd be no bulk flow from source to sink. There'd be no xylem wouldn't be moving because that's the thing that's driving the whole system. So you close your stomata, you shut down your xylem flow, you can't move anything to the rest of your body, your plant body. Having transpiration going on is what drives the entire thing. And if transpiration stops, translocation stops. they're coupled and linked together in a way that is a remarkably passive process, right? All of this flow is happening simply because of diffusion and osmosis. The only thing the plant is actually doing here in terms of what it is physically actually doing is packing and unpacking sugars into the phloem at the companion cell phloem part and moving things from the source cells to the sink cells. That's really it. This entire whole plant body process can be regulated by opening and closing stomata and by packing and unpacking phloem. That's it. Pretty awesome, huh? Plants are able to do awesome things, even though they don't have a brain, they don't have nerves, can't walk around or anything. But surprisingly, they do have some really cool behaviors that, like when you watch a plant over the course of a day, it actually moves its leaves to maintain the best angle of the sun. It's called heliotropism, right? It's following the sun. And if you're just looking at a plant, you can't even tell, right? But they're able to do it. Plants are able to recognize which way is up and down, or gravitropism. Like, why do the roots of your plants grow down and the shoots of the plant grow up? You want to hear something crazy? If you did it in the dark, the roots of the plant would still grow down. How does it know that?
What if you hung it upside down?
You hang it upside down, the roots go down. If you take one of those topsy-turvy things and flip them over, if you actually look in there and see what the roots are doing, they're going like this. They're growing, the taproot is sort of confused. It goes up and down to try and figure out what to do. Yeah, called gravitropism. How does a plant know to go to the light? Phototropism. Plants are incredibly regulatable through hormones and stuff. We're actually going to leave out the whole entire discussion of hormones here. And there's some really amazing experiments that were done, even by like Darwin and his son, recognizing that sunlight influences the release of hormones that influence the growth of the plant. Some plants even have these little things inside of them, these little cells that have a little, what we call a statolith, that's basically got a little chunk of minerals that it can tell where it's sitting in the cell, and that helps it know which way is down. I mean, really cool stuff. So, Munch's pressure flow hypothesis is one of these really awesome things about plants that help us understand how they're able to do what they do, even though they don't have the structures we see in most other organisms that make flow happen. They don't have hearts. They don't have pumping systems. They don't have muscular, dilatable, vessels and stuff like we do, or arteries. But yet they're still able to be huge and still able to have physiology that moves the things around the plant to have every cell get what it needs. So awesome, huh? You will not need to know anything about plant hormones or any what's after Munch's pressure flow hypothesis, okay? Which is not even going to discuss it. If you want to know more about this stuff, there is a class with Dr. Areola called Plant Physiology, where he goes into how hormones regulate plant growth and all kinds of stuff. So, just up through the slides, just